Hello. Let's now talk about how we discover new medicines from plants. So early on in the cycle of those lectures, we talked about the history of discovering medicinal plants by people. And that history is almost as old as the history of humankind. But we should also talk about the discovery of new plant species, because medicinal plants usually were discovered from the flora which was around people, which was growing in the region where you would live. But from the 16th century on, at least in Europe, the new era of great biological exploration have started because of the easy availability of sea voyages. And several generations of great scientists have spread around the world with a mission to discover new plant species. And to a large extent, that voyage of exploration have been driven not by a desire to discover novel medicinal species, but by pure scientific curiosity and by the growing passion for gardening, which is taking over Europe. People wanted to grow new plant species, exotic plant species in their garden. And hence a whole new generation of botanists got on the ships and went throughout the world. Famous botanists from Austria, like George Scholl, who brought amaryllis from South Africa. Probably Britain and Scotland had contributed the largest number of the eminent botanists and botany explorers. Joseph Bank is one of the greatest of them all. David Douglas, Joseph Hooker, David Lyell. Great Alexander von Humboldt from Germany, great natural historian of all times. Engelbert Kaempfer, who brought, for example, a ginkgo plant to Europe from Japan. André Town from France, worked in Paris and traveled the world, was a good friend of Thomas Jefferson, for example. And then even in the US, they had the old botanists like John Bartram, who actually was born in 1699 and lived mainly in the 18th century, followed by John Kirk Townsend, who lived from 1809 to 1851 and described a great variety of indigenous species growing in the United States. So modern age, systematic exploration of natural products, particularly those from plants, as sources of new drugs, probably started around the Second World War in early 1940s. That was just preceded by the discovery of penicillin by Fleming in Scotland. So there was this growing interest in nature as sources of specifically of medicines. One of the projects I believe really gave birth to the exploration of plants, systemic, well-funded exploration of plants as sources of new drugs, came during the World War II. Then Japan cut the supply of quinine to the US from Indonesia, where most of the quinine was produced. And the US forces were fighting in Asia, they were fighting in Africa, and they were succumbing to malaria. So US government, in collaboration with Merck and New York Botanical Garden, have started a major scientific effort within Merck to discover botanical replacements for quinine because the supply was not available. That was a major, well-funded program. And I had a great pleasure to actually know Mrs. Christine Malanga Wilson, who was one of the first women who was a director of this program because men were fighting in war. And from there on, again, more antibiotics came into being. They never actually discovered a best replacement of quinine. They had some really interesting leads, but they were never developed into drugs. And the Second World War ended, and of course, quinine became available again and then was replaced by synthetic analogs through organic chemistry. But that, to me, was one of the first examples of the systemic exploration of natural products done using the most sophisticated modern scientific research. Animal testing, they actually used young chicks to test for anti-malaria drugs. Unfortunately, the era of discovering novel drugs from natural products using modern science didn't last that long. In the 1990s, the interest in natural products and phytochemicals for drug discovery began to wane, replaced by other technology. Of course, our almost random bioexploration of plants for treatment of diseases started about 10,000 years ago. We were just randomly going around and testing different plants and observing their beneficial effects. And thousands of people who tested thousands of plants eventually produced some great information. And some of the drugs, as we discussed, which we use today, came from these bioprospecting efforts of our ancestors, which were pretty much random, not truly scientifically driven, just power of observation on their own bodies. But then about 100 years ago, chemical synthesis appeared, and people learned how to make molecules and synthesize molecules quite effectively. And with that came the pharmaceutical industry, which started to rely more and more on synthetic chemicals, in many cases actually inspired by natural products or derived from natural products, like aspirin, which gave birth to pharmaceutical industry in 1899 when Bayer took salicylic acid, a natural product from spirea, willow trees, and barks of other plants, acetylated it and produced aspirin, which is probably still one of the most commonly used drugs in the world. But then the pace and the ability to make new chemicals greatly accelerated. Combinatorial chemistry now allowed us to make thousands of different chemicals in one test tube. That was developed about 30 years ago and was used quite successfully as novel sources for drug discovery. Tens of thousands, hundreds of thousands of molecules now could be made cheaply and tested quite effectively. Screening technology evolved together with this and became high-throughput screening technology to be compatible with so many different chemicals which now needed to be tested. Target-specific computational design, or computer-assisted design, CAD, about 20 years ago, where we were able actually to model the proteins, and model the targets for drugs, and model the active sites on the protein, and using computer power, create a molecule which will effectively dock and bind the target site on the protein, therefore leading to new drugs. And then you would synthesize it, and then you test its efficacy. And of course, as we also discussed in previous lectures, the small molecule drug discovery is being gradually replaced with other ways of treating diseases, other means. And those are biologics, they're vaccines, from COVID vaccine, which is coming along as I speak, to Ebola vaccine, to polio vaccines, to vaccines which are used for cancer, to kill cancers in our bodies. And also gene therapy, where genes are being now injected as mRNAs or as modified cells, genetically modified cells in our bodies, which also help us to cure and prevent diseases. And then recombinant proteins are also, like recombinant insulin, is saving millions of lives. Anti-TNF compounds and antibodies are used to treat arthritis quite effectively, and so forth. So biologics and gene therapies probably will be more and more encroaching into the territory previously occupied by natural products and small molecules. So small molecule drug discovery from plants, while still a very, very promising area, now has to compete with many other methods of developing medicines, which is okay, because the tools with which we discover better drugs from plants are also improving, and the competition is still not lost. So to understand how we can effectively use modern science, again, to discover novel drugs from plants, we should understand the process of drug discovery itself as it now used by scientists. And there are truly two ways where you can discover drugs. One is called forward pharmacology approach, and this is when you actually have functional assays. For example, you have animals and you want to reduce you have fat animals, hyperglycemic animals, high blood glucose levels. You use those animals to model the diabetes in humans, and then you inject them with different drugs, or your phytochemicals, and you see where the levels of sugar drops. This is a functional approach. You are not really looking for a specific target. You're looking for pharmacological functional effect. Same thing can be done with cells. You can look at a rearrangement of microfibrils in the cells. You can look at the different morphological characteristics which may be associated with treatment and diseases, for example, or in humans. So this is a forward pharmacology approach, which starts with an effect, when leads to the lead compound, and after that, you actually look for which specific target this particular drug, which you discover through this functional approach, may affect. You have to identify the target because that target may be an interesting target for a particular disease. The reverse pharmacology approach is actually the reverse of the forward approach, clearly. That's where you start with the target. You start with a hypothesis. A certain enzyme, certain protein is a good, what is called druggable target for treatment of certain disease. And then you use this protein, you usually express it, make a recombinant version of it, and then do in vitro binding assays with your big libraries of your compounds. Again, phytochemicals can be one source of those libraries, or combinatorially made compounds. Or you can actually use computer-assisted drug design to make compounds which you think will bind to this particular target, and use this reverse pharmacological approach, which starts with the hypothesis a certain target is good, then finding molecules which bind to it, and then moving to the functional assays to cells or to animals or to humans to actually demonstrate that that particular molecule will lead to the desired pharmacological effect. For example, lower blood sugar in hyperglycemic animals by specifically interacting with the insulin receptor and stimulating the signaling through the insulin signaling pathway, which you already hypothesized and made certain proteins which are part of those pathways, trying to see whether binding can activate the pathway. That would be a reverse pharmacology approach. Modern high throughput approach to drug discovery, whether it's a forward or reverse pharmacology, has an insatiable appetite for compounds. Often, when you set up a high throughput screen for a certain target, for example, at a pharmaceutical company, you will only run it for several months, let's say 4 or 5 months. And you will try to use tens of thousands or hundreds of thousands of compounds in the screen, and then you move on to different target or to different function and assay. Well, clearly plant extracts are not very consistent, are not really good for that kind of high throughput approaches, just because it takes a very, very long time to prepare 10,000 plant extracts, particularly from different plants. Much longer time that the whole high throughput screen will be usually run. Also, by definition, plant extracts are complex, and they contain many different compounds which can cause non-specific interactions. That is particularly a problem in the reverse pharmacology approach, where you usually express a single protein and look for the binding, because plants make lots of compounds, just like polyphenols, for example, that can non-specifically bind to compounds and therefore interfere with the assay and hiding the specific effects of certain compounds which may be also present in the extracts. You will never see them. You'll see non-specific activity. They'll just precipitate your protein, or they'll coagulate your protein or precipitate your cells. So this non-specific binding creates a problem, significant problem. Therefore, modern approach is to fractionate your extracts by removing, for example, polyphenols from them before you will put them in the high throughput assays. Also, many plant extracts are colored, and many acid detection techniques are colorimetric techniques. So adding color to the reaction, to the well of your assay plate will interfere with the reading. You don't want usually colors there. And then you still have a challenge. Even if you have a great heat and it seems to be specific, you have to replicate your extract. You have to fractionate your extract and try to isolate inactive compounds as soon as possible. And that can take a long time. And in some cases, it's not even possible, because in many cases, well, in some cases, particularly if you use the straightforward pharmacological approach, the functional approach, you start with an assay, very often the pharmacological effect is based not on a single compound interfering or binding with your target, but a synergistic effects of several compounds which produce the desired pharmacological effect in the forward pharmacology assays. So that creates a problem, so you'll never be able to actually isolate the bioactive ingredient from your extract. Drug discovery approaches which may be more compatible with phytochemicals are actually the biorational computer-aided type approaches, like computer-aided drug design approaches. Why? Because you can do that rationally, and the structures of many phytochemicals are known. And many of those structures are very difficult to synthesize because plants are just better chemists than people are. And many phytochemicals are so complex that chemists will never synthesize them, but you can extract them from plants. And then, using the structures which are difficult to synthesize, you can do two different things. You can see whether you can virtually dock your structurally complicated natural product to a certain receptor site, so a certain target, which you know is a druggable target and maybe a good target for a drug. And then you can just isolate that compound from a plant and test it in the model system in an assay and move on to the animal systems. So that is a much more promising approach because it's a biorational approach. You don't have to deal with so many extracts. You actually will know your structure of a compound, and then if it's active, you will go to the plant and isolate the compound and test it later on. You don't have to deal with this complexity of extracts of many thousands of species. You can also, using computational approaches and algorithms, look at the structures of, for example, all existing drugs and all existing molecules which are drug-like, and try to match the structures spatially to a library of natural products. Tens of thousands of them are now available. In many cases, again, too complex to synthesize, but you can get them from plants. So if a particular drug, for example, is an analog of certain natural product which you can get from plant A, you'll go harvest the plant and extract this particular compound and then test it. So those computational type approaches, biorational approaches to natural product-based and phytochemical-based drug discovery, to me, are more feasible and tangible. It is also important to realize that in many cases, probably in most cases, when you screen the natural product libraries in your pharmacological assays, the compound which may eventually evolve as a drug will not be that particular natural product. Why? Because the drug discovery cycle, which is shown on this slide, goes as follows. You have your compound libraries. Let's say you have a library of extracts from Indonesian plants. You will put them in your primary assays, and you will look for what is called a hit, just a response. You don't know whether it's even specific. You will usually then put that extract, if you have a hit, in the secondary assays, which are more complex and more specific, and they measure other properties of your compound like bioavailability, toxicity, and metabolism. You'll study it much more, and let's say you confirm that this is a good drug candidate. That becomes a lead now. From hit, it becomes a lead. But very few of those will actually become clinical candidates right away and go in clinical trials. What will usually happen, once you know the structure, it will not be perfect in its toxicology profile, in the bioavailability profile. And what chemists in pharmaceutical companies or in academia will try to do, will try to use what is called SAR, structure activity type approaches, to improve the compound using synthetic chemistry. To attach certain groups to it to increase bioavailability. That's why halogens like thorium and chlorine, for example, are common in structures of molecule. And to decrease its toxicity. They know how to do it. They can look at the structure and they can say, if we should put this group here, it will make it more bioavailable and less toxic. So then you go through that cycle again. You synthetically make a library of compounds which are related to your lead natural product, but which have even better bioavailability or toxicological profile, and then probably better metabolism, rescreen them, and what emerges there is a synthetic compound, which is an analog of natural product, which then will go into the clinical cycle. Obviously, as we already discussed, this was done with the first ever drug which truly gave birth to pharmaceutical industry, which is aspirin. Salicylic acid was just too toxic, causes stomach ulceration, bleeding. But if you acetylate that synthetically, you get a compound which is much less toxic. Of course, then a lot of cancer drugs isolated from plants actually also made synthetically. Painkillers, synthetic opiates have mainly replaced the natural opiates now, and so forth. So this is what to be expected. Organic chemistry will contribute to the natural product drug discovery, in many cases further improving your natural product to make it a better drug. In the US, probably the largest natural product screening program was carried out by the National Cancer Institute, by its natural product branch, and it was targeting anti-cancer compound, at least in the public sector. That was the largest, and it's still ongoing, maybe at a little bit smaller scale, but they're still looking for anti-cancer compounds from natural products. And plants played a major role in this program, particularly early on. When the program started at the National Cancer Institute in the 1960s, it was done in collaboration with USDA, United States Department of Agriculture, and botanical gardens, and about 1,300 plant species were actually screened against the cancer cells, against the assays done with cancer cells, looking for specific targeted phytotoxicity in cancer cells. From 1986, this became a truly international program with about 25 different countries involved, with many agreements which have been signed with those countries, which mandated the equitable sharing of benefits if something valuable will come out of it. And almost 80,000 natural products have been screened in a panel of 60 human tumor cell lines, and most of the samples were actually pre-fractionated before. So you multiply that many samples into 10, 15 fractions produced from each of this, and 60 human tumor cell lines, you have an enormous effort. And that effort was successful, particularly actually in its early stages. And plants were mainly used because Taxol came actually as a result of this initial National Cancer Institute screening program from Pacific U. Camptothecin, which became a precursor for 2 anti-cancer drugs, also came out of this early screening programs. Compounds which were discovered later on after 1986 as a part of this international effort, and that included a lot of marine products, because marine organisms have never been looked at from the point of view of medicinal chemistry. So NCI focused on them, also focused on plants, on microbes and fungi, and many interesting molecules which are now in clinical development came out of that, but we're still waiting for a new drug like Taxol, which was a major coup and major success for National Cancer Institute. So in the last part of this lecture, I would like to introduce you to 2 important technologies which we have developed at Rutgers University, which allows fast, efficient, and very cost-effective way of collecting plant extracts and assaying them for pharmacological activity. The first collection part of this technology we call rapid metabolome extraction and storage technology, or RAMES. And second complementary technology has been designed to test those RAMES extract for pharmacological activity. And we call it screens to nature technology because it actually allows you to bring your lab to the field in the country of origin. So no samples will ever leave the country of origin and don't have to be exported to some other developed country for pharmacological testing. The strength of RAMES field collection technology is that it allows fast, efficient, and very low cost way of processing many plant extracts at the same time. It actually brings the laboratory to the field, and it allows to operate your laboratory from the trunk of your car, as you can actually see on the picture here where our graduate student, Sarah Skubal, is working on Rame's collection at Kruger National Park in South Africa. It is also very low impact. It requires minimal amount of material. It doesn't even destroy the plant. It has a high throughput capabilities. We taught this technology in about 20 different countries around the world, and this has been very effectively adapted by local scientists because they see the power of being able to go to the field and do testing there without the need of collect kilos of plant material, drying them, and then sending them to a third country for some kind of complex pharmacological analysis. So Rame's process start with collecting just 2 grams of the target tissue from your plant, which you can cut with the scissors, weigh on a portable battery-operated balance. Remember, the whole process doesn't require any external power source. All can be operated and recharged, all equipment, from the car battery. So you don't have to come back to the lab. So you collect 2 grams of tissue. You cut it into smaller pieces using scissors in a special aluminum chamber, which we supply, which costs about $3. And then the key to the extraction process is using the high revolution tool, which is called Dremel tools. You can buy it for about $30, $40 in the United States. And the only improvement to this tool which we made is to create a special blade or a set of blades, which are very effective in pulverizing and grinding plant material in a ethanolic extraction media. And we use pure ethanol to grind the plant material. We use actually 5 milliliters of pure 95% ethanol with 2 grams of tissue. So the resulting extract is about 70% ethanol, which is a very good concentration of ethanol to extract many bioactive compounds. Grinding is done in the chamber, in this aluminum chamber where I showed you. After you add the ethanol, and the Dremel tool, the rapid revolution tool, fits very snugly over the chamber using a specially designed cup, and your grinding bit almost touches the bottom. So in 20 seconds, you very effectively grind the sample. Then again, using the stainless steel sieve, which we have developed, again, very inexpensive, you filter the extract, you filter the undissolved, unextracted plant material, you filter it into basically a scintillation vial, into the 20 milliliter scintillation vial. It's very rapid. You tap on the wet extract to drain the last drops of the extract into the scintillation. And the most novel and innovative part of Rame's technology is that you can dry the extracts very rapidly by spotting them on the glass fiber filter disc, 1 centimeter in diameter glass fiber filter discs, which again are very inexpensive. And that immobilizes the extract and preserves even compounds which are rather unstable. You can effectively dry your glass fiber discs using the USB rechargeable fan in the field. Takes 5 to 10 minutes to get it to dryness, so you don't have to spend hours with a rotovap, but rather you can do it very rapidly. And now you have your discs labeled, placed in a database, and replicated 10 to 20 times, because 5 milliliters of extract and 2 grams of plant tissue will normally give you, excuse me, 20 to 30 Rame's discs, which can then be independently used for either biochemical analysis by eluting the compounds back with ethanol, or more importantly, functional analysis, pharmacological analysis in the field using screen to nature assays. This photograph shows Dr. Dushenkov, a colleague of mine, looking at Rame's samples, which we have collected at Kruger National Park in South Africa, where we worked for 2 summers looking at the natural products in the invasive plant species, again using Rame's and STN technology. We have developed a number of functional pharmacological screens which can be performed directly in the field right after you collect your Ames discs, and therefore we call them screens to nature or screens to nature technology. I don't have time to tell you about all of them. I'll just briefly mention antibacterial assay, for example, which we have developed. The question often asked is, where can you get bacterial inoculum in the field if you're not in the lab? And the answer is very simple. You can just spit in a test tube, and your saliva is a wonderful source of opportunistic pathogens, bacterial pathogens. And by diluting your saliva and putting it together with a growth media into the bottom of 24-plate microtiter dish, to which you add your Ames disc. So each well, which you see on the photograph below, has a Ames disc. And you can monitor effectively the growth of your bacteria using the live staining with MTT. If bacteria are alive, you will see the dark blue color, which indicates that your compounds are inactive. And if you see yellow color, that means that your compounds, which were on the Ames disc and subsequently were dissolved in your media, which contains some of your saliva inoculum, very effectively killed all the bacteria. So it's a very visual test. It may not be the most quantitative test, but you can rapidly identify plants which have some antibacterial activity using opportunistic pathogens and other bacteria which live in your saliva. So the STN antifungal assay uses baker's yeast as a model organism and is done in a very similar way to antibacterial assays. Baker's yeast can be purchased almost anywhere. It's used in bread baking. You add your sugar solution, which is a great media for yeast, into the well of 24 microtiter well plate. You add your Ames disc to it, and you add a little bit of yeast. You incubate for 24 or 48 hours. And then again, you add the live stain, MTT in this particular case. If yeast is still alive, the solution will turn dark blue. And if solution is clear yellow, that means that compounds on your Ames discs were quite effective in killing yeast, which, for example, is a good model organism for such important fungal pathogens like Candida. The anti-parasitic worms assay we have developed uses a very simple free-living nematode, which is a model system for roundworms, a good model for roundworms, which thrives in non-sterile oatmeal, where you can just growing its culture for months and months and months. Then you add your nematode, your Panagrellis nematodes to the well, and you can add your Ames disc again to the same wells. And you can either do live staining, or you can just visually see the nematodes. If they're dead, they'll be in a ball in the center of the well, as you can see in the well, which is top left. And if they're alive, they will still wiggle happily after 24 hours or 48 hours, depending on the temperature, depending on how exact you want to be in quantifying the effect of your compound. So we've developed over 16 different functional screens to nature assays, which are compatible with Ames discs, which are designed to work together with Ames discs, and can be deployed anywhere in the world, to the jungles, to the fields, to the oceans. They can be used with plants, but also, if necessary, with any other organism or source of natural products. And just to conclude, these are the photographs of our team working with Tajik scientists collecting Ames samples in the mountains of Tajikistan. It was a lot of fun and very productive scientifically. And this is the Ames STN workshop, which we have conducted together with our Indonesian colleagues at the Chanuan Orangutan Research Station in Borneo, Indonesia, which is an epicenter of plant biodiversity in the world. It was also extremely useful and productive and led to identification of some interesting plants with some interesting antimicrobial activities. And finally, I would like to emphasize another very important component of discovering novel medicines from plants. And this is traditional knowledge, preservation of traditional knowledge, and respect of the traditional knowledge, which accumulated over generations and generations of people living all over the world and trying the plants which grow all around them. Those are just pictures of us talking with traditional healers, traditional medicine people in Central Asia, and those people exist all over the world, and their knowledge, again, should be treated with great respect and with understanding that that represents a very valuable, precious resource which has to be preserved and maintained. Thank you very much.